digestion, you can see that acetogenesis is a process to convert all these kind of monomers and carboxylic acids to the acetate. You will accumulate a lot of acetate. However, this acetate will be quickly consumed to make the methane by the methanogenesis microbes. So in the fecal waste, if you're doing the anaerobic digestion, we see that acetate concentration is relatively low. So our idea is that if we can limit the methanogenesis process, then your anaerobic digestion process can accumulate a lot of acetate. And so we look at some references. There is a textbook by Judy Wall, and they're talking about the anaerobic digestion, and their reference report that through the AD sludge, you can actually accumulate very, very high concentration of the acetate if you can inhibit the methane production. So we look at the acetate as a carbon source. If you look at the glucose, if the one more glucose completely degrade to the CO2, and they can produce 38 ATP. You think about glucose is 6 carbon molecules. For the acetate is 2 carbon molecules. So if you think about 3 more acetate, right? So they're equivalent, carbon equivalent to the glucose. They actually can produce 30 ATP. So you can think about the acetate also have a lot of energy, almost close to the glucose, right? And so this energy actually can be utilized to make the biofuel. So the first biofuel we think about from the acetate is fatty acid. The reason is that the first metabolic step from acetate is to synthesize acyl-CoA, and acyl-CoA is direct precursors of the fatty acid. So we did some calculation. Our calculation show that 1 gram acetate actually can produce about 0.29 gram fatty acid. You know that fatty acid is the type of the biodiesel precursors, right? You can use fatty acid to produce a biodiesel. So now we think about whether we can do the biodiesel from this acetate with acetate. So we did some anaerobic digestion of the fecal waste. And so this part of work was done by Michigan State, my colleagues. And so they tried a different method because accumulated acetate, there's a lot of literature shows that, such as change the pH or temperature, or adding some air, or addition of the chemical inhibitors. So they tried some tests, and they found that actually acetate can be accumulated by adding some chemicals called the iodoform. At a room temperature, we can see that if you do not add the inhibitors, you only get about 1 gram to 2 gram acetate. But if you add this inhibitor, the acetate production is increasing. However, I want to emphasize that this experiment is not optimal, because we saw the reference if people doing the thermophilic anaerobic digestion and you add some inhibitors, the reported data is 40 grams per liter acetate. So again, we still have a lot of room to improve this process to make the acetate. So since we have the acetate, and so our colleague, my friend, he tried to use some natural fungi to utilize acetate. So this bug, modular, it is actually a very strong bug, and they can they report they can use acetate very well, and they also can accumulate the biodiesel in their lipids. So he used this waste acetate to go in this bug, but the result is pretty disappointing, because we only had the best result. We only have about 5.8% conversion of the theoretical yield. So the lipid content is very low if you use a natural bug. So in my lab, we are thinking about using some metabolic engineering to actually make the microbes more efficiently to convert fatty acid from the acetic acid. So our work actually follows the standard genetic engineering method. First thing is that we have to overexpress the gene for the acetate utilization. So especially the ACS gene, the acetyl-CoA synthesis is very efficient, make the bug to eat the acetate very quickly. And then we also try to knock out the acetate production pathway from the E. coli, and we overexpress two key genes for the fatty acid synthesis. And we also knock out the fatty acid degradation pathway. So we did some genetic modification using E. coli. You know that E. coli is also present in the fecal waste, right? So the engineered E. coli, we found that if we overexpress TES-A gene, which is a very important step for fatty acid production, this gene encoded the enzyme called the acyl-ACP. They actually can give the highest fatty acid production. So all these type of the 4 genes have been recently reported by other reference. I want to show you the fatty acid production. Research is very, very hot in the biofuel field currently, so we can easily get this type of reference, and we tried this type of work based on reference, and we did the work in our lab, and we actually selected the TES-A gene as an optimal fatty acid production loops. So we construct several mutants and we look at their fatty acid production from the acetate. So that experiment is pure acetate work, so there's no anaerobic digestion involved. So we found that if we just overexpress ACS gene, which is acetate utilization pathway, and the TES-A gene, which is the fatty acid production gene, we can get actually about 0.36 gram per liter fatty acid using acetate. But that acetate concentration inoculation is relatively low. I think we just use a few grams acetate. If we did more work, we put 3 gene modification, we knock out the fatty acid degradation pathway, we can further improve the strain performance to make the fatty acid. So we did some fermentation, and this fermentation experiment using the pure this acetate, and this fermentation control is actually coupled with the pH meter. So we control the pH as neutral, and because once the acetate you use, then the pH will be increased, right? And we add more acetic acid to balance it, so the cell can utilize acetic acid very in an optimal condition. So we can see this is the growth curve and the fatty acid production curve. So that the result shows that if we the median is a rich median, so that we have some yeast extract inside. For example, like the fecal waste, they have amino acids or other carbon salts, right? The yeast extract is similar thing, so you have a lot of different carbon salts. And the cell can grow very well, and they actually can make about almost 1 gram per liter fatty acid. However, if you look at the second figure, and they show that if you use a minimal media, so you only have acetate as the carbon salts, the cell usually grow with a very long lag phase, and they also produce less, only half of the fatty acid. So I think the nutrient source is also very important for this fermentation. So basically, our experiment observed that fatty acid production is growth associated, and yeast extract promoted growth and fatty acid production. So now, since you use a yeast extract, so we want to do a double confirmation that how much yeast extract contribute to the fatty acid production. So we did some C13 labeled experiment. So we labeled the acetic acid with carbon C13, and so there is a non-labeled carbon nutrients present in the culture, right, the yeast extract. So once the cell metabolizes these carbons, and you can see that we harvest the biomass, and we look at their metabolites. So we derivatize them, we use GCMS to do the measurement, and we analyze their isotopic distribution. And so you can see that if the metabolite is from the acetic acid, so you will see these metabolites will be labeled, right? And if the metabolite is from the unidentified carbon nutrients in the yeast extract, then they will be not labeled. So our experiment shows that, okay, if you have a leach medium and our mutants actually do not actually utilize acetic acid too much to synthesize the biomass. If you look at this curve, this is mean C13 abundance. So that means how much percentage of the acetate are incorporated into the biomass. This biomass we are looking at just amino acids, and we see that most amino acids are unlabeled, right? So labeling is only 10% to 40%. So that means the cell actually like to uptake other type of nutrients instead of acetate. But acetate is not a very energy, not a very metabolic favorable substrate. However, if we look at the product, the fatty acid product, then this is a mass spec peak which shows, and the first figure shows the fatty acid, non-labeled fatty acid profile. Then the second figure which shows a labeled fatty acid profile. So after the fermentation, and you see the peak have a strong shift from the labeling part. So we did a calculation based on this isotopic balance. So we look at some characteristic peak, and we did some calculation using this equation, and we found that actually average about 63% of this fatty acid, so the fatty acid carbon, from the acetate. So that means that after genetic engineering, the cell actually utilized majority of this acetate carbon to make the biofuel fatty acid. So that is good news. We like it. And so we did a comparison. So we grow this the culture. So first thing is that we grow the two different one is a natural the fungi species, which we know that they accumulate the biodiesel, and the other bug is E. coli. We engineer them, and so we grow them using two different medium. One is pure acetic acid. One is we got the medium from the anaerobic digestion, and which is we limited the methane production. We accumulated the acetic acid. So we can see that if we compare these two species, the engineered microbes have much higher performance than the natural wild type fungi species. So that means we can use genetic. This proves that we can use genetic engineering tools to improve the biofuel production from this waste acetate. And also, we look at this conversion yield. And if we use the AD waste, our calculations show that we have about 31% of the yield of the theoretical yield. So that is higher than the wild type species. We believe that the cell not only utilize acetate, they probably AD also have other nutrients, and so that helped improve the yield of this biofuel. So most exciting result is that if we look at the fatty acid profile, and if we use the E. coli to grow the grow on this AD waste or acetic acid, so the chain length of the fatty acid is much shorter than the normal fungi species. Or we know that the biodiesel from plant or from the animal fat, they are pretty long. They are 18 or to 22 carbons. So you only can use a biodiesel. But our majority carbon length is actually falling to the median chain of fatty acid length, which can be utilized as a jet fuel. Because jet fuel, if your carbon number is below 14, and then people found that this type of the biodiesel will mix with the jet fuel and use it as a jet fuel to fry your boiling pan, right? So that is another exciting result, that we actually can convert potentially I can say that convert the waste, fecal waste to very advanced biofuel. Okay, so again, the acetic acid not only came from the fecal waste. So we did some literature search. So if you have a microbials, they can convert the acetic acid to biofuel, and you can actually not only get the acetic acid from the waste, the fecal waste, because organic waste, anaerobic digestion, you can make the acetic acid. Also, in the United States, we have a lot of methane, natural gas in Pennsylvania. And this methane can be easily chemical conversion to acetic acid. Then you use acetic acid to make the biofuel. And another type of the acetic acid that comes from is first you can from also fossil fuel. So say that you make the syngas from the coal, right? You also can make the syngas from the lignocellulose by the gasification. And then this syngas can also convert to the acetic acid through the chemical catalyst, chemical conversion or microbial conversion. And also that if you look at the cellulosic ethanol industry, so if people want to convert the lignocellulose to biofuel or bioethanol, you have to do a pretreatment. The pretreatment is that you hydrolyze the lignocellulose to sugar. During this process, you will also produce a lot of acetate. But this type of acetate, probably people don't like to use it because they are inhibitory to the fermentation. People want to wash them away. But we actually can use this verb to also utilize this waste acetate from this lignocellulosic bioenergy industry. So that indicated the application. You have a lot of different angles. So again, we can see there's a lot of advantages for this using acetate as a carbon source because they're cheap and plentiful, and they have enough energy to make the biofuel, and they actually also, since the time is limited, I don't have time to talk about the second work, is that we also tried to engineer the cyanobacteria and with isobutanol pathway to convert the CO2 to the isobutanol, which is another biogasoline. And that also tells you that through the anaerobic digestion, the CO2 also can be potentially recycled and captured, make the biofuel. This work I will not talk about since the time is limited. But so the final goal, this is what we think about the potential of this project is that we can use anaerobic digestion. We inhibit the methane production. We accumulate acetate, and we can integrate this process with solar energy this equipment. So we can use the solar heat to pasteurize the AD, the effluent, and then the acetate can go feed with the fermentation tank, and then we can make the biodiesel. We can also think about engineering yeast, which is that which can efficiently produce the butanol. Yeast also like to grow in the acidic pH so that we have better performance than coli to produce the biofuel. That is will be a future work. And also, we are thinking about trying to capture CO2 and anaerobic digestion have a lot of nutrients we want to actually energy process to retain the nutrients in other biofuel. So one thing is when I talk about this process, other people think that concern say that worry about that. You use a genetic engineer the bug, and they may have some potential environmental impact. So okay, then in this case, what we can do is that we remove this algae, genetic engineered algae, replaced by some biofertilizer algae, so such as microcolores. And this type of blue-green algae, they can capture the nitrogen source, and they can also be used as a biofertilizer. So that is very useful, but can help the soil. So the lastly, and yesterday I fly with a professor from Delaware, Dr. Dental, and And so he also told me that he have a very nice hydrophobic membrane, and this membrane can be applied to capture acetate directly from the anaerobic digestion tank. So that if this is the case, then that's very good for this process, because you can get the pure acetate for fermentation, and you can limit this toxic chemicals and still stay in the anaerobic digestion tank. So our future work has 3 parts. So enhance the anaerobic digestion acetate accumulation, and increase the metabolic conversion, and improve recovery and water reuse. So this is our acknowledgement. So when I first started this project 1 year ago, I have no experience on the fecal waste, so I have to talk to my friends. So this project create the opportunity for me to work with other universities. So I'm also very, very willing to use anyone interested in this, and you can talk to me and we can work together. So I'm a metabolic engineer, and I like to work with environmental engineering people. And so, the Michigan State University helped me to do some anaerobic digestion work, and I also have a colleague at the University of Washington. We are in the same lab, postdoc lab, and James Carlos, and he's helping me on some molecular engineering work. So the project is supported by the Gates Foundation. So again, thanks for all this.